Good morning, ladies and gentlemen. I'm Sabah Riaz, PhD scholar from Department of Zoology, Government College University, Faisalabad. I warmly welcome all of you on the 7th International Conference on Applied Zoology 2024. I will be the moderator of the session, which will include from the field of biodiversity, entomology, pest management, and toxicology. Let's start our session with the talk of our first speaker, Mr. Ubaidullah Kaima from Mila University. He will be talking the Beni Harmonda as critical wintering site for western Palearctic waterfowl, an ecological survey and inventory, Mila Northeast, Algeria.
Good morning, everyone. I just want to thank you for giving me the opportunity to participate at the 7th International Conference on Applied Zoology. So, first of all, I want to introduce myself. I am Ubaidullah Kaima, a PhD student at the University Center of Mila, Algeria. In this presentation, we're gonna talking about the Beni Haroun Dam as a critical wintering site for Western Palearctic waterfowl, an ecological survey and inventory, Mila, Northwest Algeria. In the introduction, we're gonna say that in Algeria, the ecology of water birds, their migratory pattern, wintering behaviors, and activity rhythm have been extensively studied in the several natural wetland area, but have been largely overlooked in artificial wetlands. The Beni Haroun Dam is a vast strategic hydraulic complex in Algeria, considered the largest dam in terms of surface area nationally. This picture represents the location. This picture represents the location of Beni Haroun Dam in Algeria. We're gonna move to the material and the method. Three study sites provide an adequate view of the birds on one hand, and on the other hand, a sufficient view of the entire dam. The avifauna was monitored twice a month at all study sites. We used a different observation point at each station. The counts are conducted by direct observation of birds using a binocular, a telescope, a camera, and the Hansel guide. We're gonna move to the results and discussion. Here we have a picture number 2 represent special occupancy of the Beni Haroun Dam by Arditi. As we can see here, we have the special occupancy of the Beni Haroun Dam by Arditi in the three sites. The first all is Al Maleh, the second is Al Medus, and the final set is Douar Al Bedi. We're gonna move to the picture number 3 who represents the special occupation of the Beni Haroun Dam by Ralidi at the three different sites.
Thank you so much, Libedla Kaima, for sharing your research work. Now, moving forward, I would like to invite our second speaker of the session, Saif Eden Marzong from University of Gulma. He will present his research work, The Importance of Artificial Wetlands in the Mala region for the Wintering of Birds, Water Birds, the case of Sidi Khalifa Dam.
Good morning, everyone. I want to thank you for giving me the opportunity to participate at the 7th International Conference on Applied Zoology 2024. So, I am Labat Lajaima from the University Center of Mila, and today I will be presenting the results of a study on the importance of artificial wetlands in the Mila region, focusing on the wintering of water birds with the Sidi Khalifa Dam as a case of study. Wetlands are ecosystems located between land and water. They are among the most productive natural environments in the world. Biodiversity provides water and the primary resource upon which countless plants and animals depend. Artificial wetlands are freshwater wetlands area created as a result of human activities such as dam. Algeria is rich with approximately 1,700 of them. These are either artificial seats such as dams or natural ones such as rivers. Water bird population use wetlands during one or more phases of their biological cycle. They are utilized differently by this population, depending on their ecological recruitment and phenological states. Let me first provide a brief overview of the geographical situation of the Wilaya of Mila and the Sidi Khalifa Dam, which is the focus of this study. The dam is an important site for migratory and wintering water bird species. This picture represents the geographical situation of the Wilaya of Mila in Algeria. And this picture represents geographical situation of the Sidi Khalifa Dam. Our study involved twice monthly monitoring at the 3 selected sites using a binocular, a telescope, and a camera. We conducted direct observation of the birds assisted by Hansel's guide for identification. Then we have the results and discussion. We observed 29 species across 12 families, including Anatidae, Ardidae, Rallidae, Laridae, and others. Let me walk you through the key finding. As shown in the P chart, Rallidae made up the largest proportion of wintering species, accounting for about 42% of total, while Anatidae and Laridae followed at 16 and 17 respectively. This demonstrates the significant presence of Rallidae in the Sidi Khalifa Dam, which could be due to the dam's suitable habitat structure for species. Here we have some pictures represent the different species of Anatidae. There we go here. We tracked the population of cave species across the month from October 2023 to March 2024. For example, in the Anatidae family, the mallard, the canard calvaire, population peaked in January 2024 with 403 individuals before declining in February and March. This is indicative of the species' migration and adaptation to seasonal change. The fluctuation in number reflect the migratory patterns of the species, which are influenced by the availability of food and shelter at different times of the year. About the family of Possibilidae, the population peaked on December 1 by 401 individuals. The RDD peaked on October 2 by 121 individuals. The Ciconidae peaked on March 2 by 32 individuals. The Rallidae peaked on October 2 by 1,427. The Laridae peaked on October 2 by 514 individuals. The Phalacrocoracidae peaked on January 1 by 15 individuals. The Phaneroceptiridae peaked on February 1 by 6. The Gryidae peaked on March by 10 individuals. The Cardiidae peaked on January 2 by 19 individuals. The Scolopacidae peaked on February 1 by 14 individuals. And the last family peaked on February 1 by 28 individuals. At the end of our study, we highlighted the importance of artificial wetlands and their role in the environment. These environments are exponentially rich on biodiversity, as they modify the hydrological system at all levels within species, affecting their decline or disappearance. In light of the results obtained from the study conducted at Sidi Khalifa Dam, we recorded 29 species belong to 12 families. The most represented family is the Ardiidae family with six species. The protection of artificial wetlands could significantly contribute not only to the maintenance of birds, but also to that of many other elements of biodiversity. I want to thank you for your attention.
Thanks a lot, Safin Edin, for your wonderful talk. Now, I would like to invite Mavish Salman from Government College University, Faisalabad. She will talk on the bioactive metabolites of Cocoria oceani exhibited antibacterial, antioxidant, and antidiabetic activities.
Bismillahirrahmanirrahim. First of all, I would like to thank the organizers of 7th International Conference on Applied Zoology 2024. I am Dr. Mavish Salman, working as Assistant Professor in the Department of Biochemistry. The title of my presentation is Production of Bioactive Metabolites from Cocaria species for Antibacterial, Antioxidant, and Antidiabetic Activities. These are the contents of my presentation. First of all, a brief introduction about my presentation. The secondary metabolites are bioactive organic compounds that inhibit the growth of microorganisms, and these bioactive secondary metabolites are natural products that show antibacterial activity against multi-drug resistant pathogens. Actinobacteria. The Cocaria species belong to actinobacteria that produce various types of bioactive secondary metabolites. These metabolites have the potential to combating a variety of pathogenic microorganisms. The aims and objectives of my study is extraction of secondary metabolites from Cocaria oceani, determination of bioactive potential, including antibacterial, antioxidant, anti-inflammatory, antidiabetic, identification of secondary metabolites that have potential bioactivities present in Cocaria oceani by using various spectrometry techniques. This is the scheme of the study. First of all, we grow bacteria in its respective medium for the production of BSMs that are bioactive secondary metabolites. Then we extract these BSMs by using appropriate organic solvent. Then various bioactivities were performed, including antibacterial, antioxidant, anti-inflammatory, antidiabetic, and antilipidemic assay. And beside this, these bioactive BSMs were identified by using GCMS and electrospray ionization mass spectrometry. These are the methodologies which we have followed. This is the culturing of the bacteria, and after having growth on Petri plates, we scrap or all the growth and then process it further for the extraction of bioactive secondary metabolites by using ethyl acetate as a extracting solvent. Then we performed rotatory evaporator for to concentrate or to evaporate or solvent. Now we move to results. These are the results of antibacterial assay of BSMs. We have used 4 strains: Staphylococcus aureus, Escherichia coli, Bacillus cereus, and Serratia marcescus. And these are the inhibition zones measured in mm. These are the results of antioxidant assay of DPPH scavenging assay. It is a dose-dependent assay, and we have maximum concentration of inhibition activity of 83.87% at 600 microgram per ml of our BSM. This is another antioxidant assay, hydrogen peroxide scavenging activity, and it is also dose dependent. And we found maximum percentage of hydrogen peroxide scavenging activity at 600 microgram per ml, and its percentage is 91.93%. Next is the anti-inflammatory activity, which was performed by albumin denaturation, and is also dose dependent, and we found maximum concentration, percentage of inhibition at 600 microgram per ml. It is another anti-inflammatory assay which is heat-induced hemolysis. It is also dose-dependent, and we obtained maximum inhibitory effect of hemolysis at 600 microgram per ml, and its percentage is 81.39. Anti-diabetic assay. It was performed by alpha amylase inhibition, observing alpha amylase inhibition in percentage. It is also dose dependent, and we got maximum concentration, maximum inhibition of 90.97 at 600 microgram per ml. This is another anti-diabetic assay, that is glucose uptake by yeast, and these 92.77% is the maximum effect which was recorded at 600 microgram per ml. Next is the anti-lipidemic assay. This anti-lipidemic assay was performed by observing the percent inhibition of pancreatic lipase, and its maximum value is 76.73% at 600 microgram per ml. Now we move to the identification of bioactive metabolites. First, we have identified various volatile metabolites by using GCMS, which have various bioactivities. These are the names of the identified compounds with respective retention time and concentration and peak area. We have identified 15 compounds with the help of NIST library. And these are the nature of these compounds with molecular formula, molecular mass, and bioactivities. And we observed that most of the compounds have antimicrobial, antioxidant, hypotensive, anti-cancerous activities, and anti-tumor activities, anti-proliferative activities. These are the activities of compound which we have obtained or identified by GCMS. These are the compounds which were identified by electrospray ionization mass spectrometry. The identified compounds were succinic acid, glutamic acid, maglutol, caffeic acid. These next in the next column, these are the MZ values, molecular mass, and molecular formula of these compounds. Among these compounds, the maglutol is a drug which is an anti-lipidemic drug, and we have identified this drug from Fucaria species first time. This one is the spectrum of ESI MS2 of succinic acid in negative mode, and along with the MZ values. And this one is the spectrum of glutamic acid and maglutol, caffeic acid in negative mode with the MZ value of 179, and along with their structures. To the best of our knowledge, identified bioactive secondary metabolites have not yet been reported from Fucaria oceani earlier, including the drug which is maglutol. Now, I would like to conclude my research work. This work represents that Fucaria oceani bioactive secondary metabolites had potential antibacterial, antioxidant, anti-inflammatory, anti-diabetic, and anti-lipidemic effect. Further research is needed to clarify their precise mechanism of action and determination of most effective therapeutic application of BSMs of Fucaria oceani. Thank you so much for your kind attention. Thank you.
Thank you so much, Mavish Salman, for your wonderful talk. Now, I would like to invite Talha Riaz from Ozang Agriculture University, Wuhan, China. He will present his research work on the zooplankton biodiversity's role in nutrient cycling and stability in aquaponics.
Hello everyone, my name is Talha Riaz, and I'm here at the 7th International Conference on Applied Zoology, which is organized by University of Sargodha. I think this conference is a wonderful opportunity for me and the other speakers like me to explore the application of zoological sciences for sustainable development and the practices which are taking place all around the world. I'm basically from College of Food Science and Technology, Hwa Chong Agricultural University, Wuhan, and I am doing my PhD in food science and working in the area of alternative proteins, especially the microbial proteins. So here is a basic overview of aquaponics. Aquaponics is basically a sustainable method that combines aquaculture, in which we raise fishes with hydroponics and by growing of plants in water. And this system not only recycles nutrients and water, but also creates a self-sustainable ecosystem. For example, fish waste serve as a natural fertilizer for plants, while plants help to purify the water for the fish. This relationship has become significantly reduced the need for chemical fertilizers and conserves water, which is the basic need of today. We talk about the importance of zooplankton in aquaponics. Zooplankton play a very important role in the ecosystem. For example, they act as the bioindicators of ecosystem health, and their presence and delivery can signal the overall well-being of aquaponic system. And they can also contribute to the nutrient cycling, for example, enhancing the water quality by breaking organic material and replenishing vital nutrients. If we see the nutrient cycling mechanism, we can see that the rotifers, for example, efficiency break down organic waste from the fish. This process increase the bioavailability for essential nutrients like nitrogen, phosphorus, and they are very crucial for plant growth. By improving nutrient cycling, zooplankton help maintain a balanced environment where both fish and plants can thrive. An environment where fish is working for the plants and the plants are working for fish. Then I will be talking about the impact of phytoplankton population. Copyponds regulate the phytoplankton population and they help to mitigate the risk of harmful algal blooms that can deplete oxygen levels in the water. And they can also promote a balanced microbial ecosystem which support overall plant health in aquaponic system. The presence of diverse species like zooplankton species, they can help remain stable and healthy. Due to the presence of this large variety of organisms, it is a sign of a stable and healthy ecosystem. If we talk about the biodiversity and system resilience, diverse zooplankton communities contribute to reduce the ammonia toxicity and they enhance system resilience. These communities are better equipped to withstand fluctuations in the water quality, temperature, and they make the aquaponic system more robust and sustainable over the period of time. Biodiversity act as a buffer against the environmental stresses. If we increase the biodiversity, the environmental stresses and the environmental harmfulness factors will automatically be reduced. If we talk about the management practices, despite their importance, effective management practices for maintaining zooplankton population are still very underdeveloped. For example, there is a pressing need for targeted research in this area. Develop strategies for habitat enhancement and population monitoring can ensure that zooplankton communities remain healthy and functional. Now we will see what are the future research directions and what are the research gaps that we can see in this area. Future research should focus on methods to improve the zooplankton diversity through habitat enhancement and nutrient management. Both the habitat enhancement and nutrient management are very crucial factors to maintain a proper ecosystem in which zooplankton play a very important role. By optimizing these populations, we can significantly increase the aquaponic system productivity and contribute to the sustainable food production initiatives. Like the issues which the uh, modern world is facing, like food security and other issues that can be uh, solved with the help of these if we maintain a proper balance in the ecosystem. Collaborative efforts between researchers and practitioners will be the key to these advancements. I would like to say that there is a need of multidisciplinary research in which researchers from the different backgrounds, they work together, they think together, and they invent new techniques by which there is a stability between the ecosystem in which zooplankton diversity can play a very important role. So, what are the key takeaways from this presentation and from this topic is that zooplankton are integral to nutrient cycling and they maintain stability in the aquaponic system. Prioritizing their biodiversity in future research is very important for enhancing productivity and sustainability. As we continue to explore the potential of aquaponics, we must recognize the vital role that those small organisms play in achieving our environmental goals. So that's it from my side. Thank you so much for your attention, and I welcome any discussions at this time. And I really look forward to your insights and thoughts on this important topic. Thank you so much.
Thanks a lot, Talha, for sharing your research work to our audience. Now, I would like to invite Raza Muhammad from Kolakong University, Bangkok, Thailand. He will be talking on the gut microbiome of long-tailed macaws, a nexus of environment, diet, and host genetics.
Greetings of the day. My name is Raza Muhammad, and I'm a PhD candidate at Chulalongkorn University in Bangkok, Thailand. Today, I would like to talk about the gut microbiome of long-tailed macaques, a nexus of environment, diet, and host genetics. Macaca fascicularis, or long-tailed macaque, is one of the non-human primates that is widely distributed throughout Southeast Asia and is currently being used in biomedical research. Based on morphological characteristics and geographical distribution, this species has been divided into 10 subspecies. Among these 10 subspecies, Macaca fascicularis aurea, or MFA, is strikingly important because of their stone tool use behavior during foraging to access in-case food items such as oysters or nuts. MFA originates in Myanmar and is distributed southeastwardly in Thailand through the Mergui archipelago. MFA lives in close contact with another subspecies of long-tailed macaque, that is Macaca fascicularis or MFF. However, MFF has never been reported to use stone tool in nature nor in captivity upon training. Thus, previous studies indicated that distinctive genetics characteristics between the two subspecies. Thus, it has been hypothesized that different genetics might be one of the factors that led to the emergence of stone tool use in MFA, but not in the other subspecies. Moreover, MFA primarily inhabited coastal and estuary habitats to a greater extent than the overall MFF populations. With respect to the stone tool use behavior being found only in MFA, it has also been proposed that it might be due to the differences in their habitat types, which in turn could reflect the different types of food availability in their natural habitats. Therefore, we hypothesize that variations in the microbiomes between the two subspecies of MFA and MFF might correspond to their habitat type and dietary preferences in association with the performance of their stone tool use behavior. This lead to the research question that do differences in bacterial microbiomes between MFA and MFF relate to habitat, dietary preferences, and their stone tool use behavior. Two populations, each of MFF and MFA, residing on the island and in mangrove forest, were selected, as you can see in the photo. And the food types that were consumed by the monkeys were identified either by direct observations or by leftovers of food items, which the monkeys finished eating. Fecal samples were non-invasively collected in the field by fecal swab method, as you can see in the photo, and the DNA was extracted and the full-length 16S rRNA gene was amplified and finally sequenced on the MinION platform. The results showed that the two populations of MFF were receiving anthropogenic food from tourists and local people, while the MFA populations were solely relying on their natural habitats. Based on the diversity analysis using Chow-1 and Shannon indices, it showed that the MFF populations who were receiving anthropogenic foods in addition to their natural food exhibited higher alpha diversity as compared to the MFF populations, while the beta diversity showed that the MFA mangrove population exhibits distinct bacteriomes. Now here a question arises that why do the alpha diversity is higher in MFF populations than MFA population? And it can be seen in a previous paper that has also reported that the provisioning leads to the higher alpha diversity. And it might be due to the introduction of the new foods that apart from the natural foods in their natural habitats, these provision macaques could also access additional food regularly provided by humans, and it could possibly lead to the higher diversity of their microbiome. Based on taxonomic classification, the top taxa at the phylum level were Firmicutes and Bacteroidetes, while the genus, the macaques, were highly enriched in Prevotella, which lead to the carbohydrates and the fibers. While at the species level, these populations show the higher diversity of Clostridium difficile, which can lead to the infection of Clostridium difficile by the Clostridium sensitis taxa. Moreover, the left-C analysis revealed that the MFF island population exhibits higher bacterial taxas as compared to the other populations of the MFF and the MFA at mangrove and island populations. To conclude, these results suggest that the diet, which includes the natural food, anthropogenic food, and the foraging behavior, which is the stone tool use behavior, and also the host genetics, because the two subspecies have distinct genetics. And the third one is the environment, which lead to the differences in the habitat types have an influence on the gut microbiota of these long-tailed macaques. And as I mentioned, that the two populations were receiving foods from the humans. One of them were receiving from the tourists, because some of the populations are living on the islands, which is an tourist attraction for the tourists, which regularly feeding by the humans, and also the local population also feeding the monkeys, which leads to the macaque dependence on their provision food. Thus, it has been advised and suggested that the monkeys should not be feeded in their natural habitat because it could affect their natural behavior. And finally, I would like to acknowledge all the people who supported and contributed in this work, and thank you for your kind attention.
Thanks a lot, Raza Muhammad, for sharing your research work. Now, I would like to invite our last speaker, Mr. Muhammad Abdul from University of Education, Faisalabad. He will be talking on the evaluation of essential and toxic elemental levels in the tumor and non-tumor tissues with risk of colorectal cancer.
بسم الله الرحمن الرحيم I am Dr. Hafiz Muhammad Abdul Qayyum from University of Education Lahore, Faisalabad campus. The topic of my presentation is patterns of essential toxic metals distribution in the hair of lung cancer patients in comparison with healthy donors. First of all, introduction. Cancer. Cancer is not a single but group of diseases characterized by uncontrolled growth of cells and impaired cellular maturation. Cancer develops only in cells with damaged genes. It is a multifactorial and multi-mechanistic disease that represents a great variety of malignant tumor which can affect any part of the body. Cancer is the leading cause of death in economically developed and second leading cause of death in the developing countries. Almost 1 in 4 deaths were reported due to cancer in the world. In America, a total of 1.7 million new cancer cases and 0.5 million cancer deaths were reported in 2019. Whereas in Pakistan, an estimate of 0.5 million new cancer cases and 0.1 million cancer deaths were recorded in 2019. In worldwide, 14.1 million incidents of cancer and 8.2 million deaths due to cancer were estimated in 2019. According to WHO, by 2030, almost 22 million people will die from cancer all over the world. The stepwise development of cancer involves different stages, including initiation, promotion, progression, and metastasis. Initiation is characterized by genomic changes within the cells, such as point mutation, gene deletions, and chromosomal rearrangement. Whereas promotion, the tumor development is promoted by survival and clonal expansion of these initial cells. In progression, consists of substantial growth in tumor size, whereas metastasis, cancer cells travels to other parts of the body where they begins to grow and form new tumors. Lung cancer, cancer that forms in the tissues of the lung, usually in the cells lining air passages such as bronchioles or alveoli, and disrupts the proper function of the lung. It is number one killer in men and second in females. Overall, 1 in 17 deaths were reported due to this cancer. In worldwide, 3 million new cancer cases were estimated to arise. In Pakistan, around 20,000 new lung cancer cases diagnosed in every year. 5-year survival rate is only 14%. Types of lung cancer. According to WHO, lung cancer is classified as 2 major histological types. Number 1 is the non-small cell lung cancer and second one is the small cell lung cancer. Each type of lung cancer grows and spreads in different ways and is treated differently. Stages of lung cancer. Stage refer to determine how far the cancer has spread in local tissues and other sites of the body. These are used to develop a treatment plan and to define patient prognosis. According to World Health Organization, lung cancer is divided into 4 stages, stage 1, 2, 3 and 4, whereas stage 1 is the slow growing and stage 4 is the most advanced or highly aggressive. The risk factors which are involved to spread in lung cancer: tobacco smoke, crystalline silica, asbestos, radon gas, genetic factors, radiation, immune conditions, diet low in fruits and vegetables intake, and metal imbalance are the most important risk factors of the lung cancer. Significance of hair samples. Scalp hair resides in the dermal layer of the skin and is made up of 20 different cell types, each with distinct functions. The affinity of metals for hair is due to the presence of cysteine in the keratin structure as well as to follicular melanin, which are able to bind cations by ionic interactions. Concentration of metals in this matrix reflects from its mean level in human body during a period of 6 to 12 months as compared to other biological samples. Human hair has many other attractive disadvantages such as finer stability, easy collection, transportation, convenience to reflect long-term exposure to high metal, and high metal concentration than in other bioindicators. Significance of selected metals. Essential metals are required for normal growth and development of human body, while their deficiency lead to various ailments. Therefore, metals play both curative and preventive role in combating disease. Trace metals are required at optimum concentration for proper function of the human biological systems. Calcium and magnesium are vital elements for skeletal formation and regulate heart rhythm and immune function, whereas cobalt is an integral component of vitamin B12, takes part in RBC function formation. Cadmium is a non-essential for human. It induces nephrotoxicity, immunotoxicity, and osteotoxicity. IARC classified cadmium as a group 1 human carcinogen, develops lung and prostate cancers. Chromium is an essential trace element required for maintenance of glucose and fat metabolism. Copper occurs in more than 300 enzymes. Its toxicity may lead to lung cancer, oral cancer, and ovarian cancer. Iron is mostly utilized in hemoglobin. Its toxicity typically damages cells in the heart. And manganese is necessary for brain and skeletal development. Sodium and potassium both work as a body electrolyte and control the transmission of nerve impulse and muscle concentration, contraction, and relaxation. Nickel is an important human urgent and develops inflammation, bronchitis, cardiovascular, and kidney diseases. Lithium is very important in psychiatry because it acts notably at the level of neurotransmitters. Zinc is an antioxidant and anti-inflammatory agent. Strontium promotes strong osteoporosis, resistance bone, and reduce the pain of bone lesions. Lead toxicity may induce anemia, kidney damage, neurological effects, hypertension, and reproductive disorder. The association between occupational exposure to lead and lung cancer is well documented. Stress and cancer. Redox active metals undergo cyclic reaction, participating in the transfer of electrons between metals and substrates, and play an important role in the maintenance of redox homeostasis. Free radicals are known to react with all components of DNA, thus damaging its bases and deoxyribose backbone, causing mutation in crucial genes. Process of breakdown of metal ion homeostasis due to oxidative stress involved in a plethora of diseases such as cancer. Following figure clearly demonstrated the mechanism behind the development of cancer. Major objectives of the present study: to examine the comparative distribution of selected metals in the hairs of cancer patient and healthy subjects, to investigate the mutual relationships among the metals in terms of correlation coefficients, to evaluate the comparative apportionment of essential toxic metals, to investigate the viable variations in the metal levels with food, with habitat, food, and smoking habits in the hairs of cancer patient and healthy subjects, to determine the significant differences in the metal concentration in types and stages of lung cancer. Experimental methodology consists of following 4 steps: sample collection, sample preparation, metal analysis, and the last one is the statistical analysis. Collection. The hair samples were collected on volunteer basis from the newly diagnosed cancer patient admitted in Punjab Institute of Nuclear Medicine, Peenam, Faisalabad. Samples from the healthy subjects were also collected. Information regarding donor's diet, age, abode, smoking habits, health status, medication, occupation, cancer type, stage, etc. was recorded on a pro forma. Sample preparation. First of all, washing of hair samples. The samples were cut into small pieces of 2 to 3 cm length and mixed with 50 ml of detergent solution in a conical flask. The samples were washed with plentiful tap water. It was followed by addition of 30 ml of treated standard solution and again shaken on an auto shaker for 30 minutes. It was finally rinsed with double distilled water. So the acid digestion of hair samples, the digestion was carried out on hot plate. An accurately weighed portion of the hair sample was treated with 10 ml of concentrated nitric acid and heated at 75 centigrade for 40 minutes. It was cooled to room temperature, followed by addition of 5 ml of perchloric acid with subsequent heating to soft boil until white dense fumes evolved, marking the completion of the digestion process. Metal analysis by flame AAS. Following selected metals were analyzed by flame atomic absorption spectrophotometer, implying optimum analytical conditions. This table illustrates the optimum analytical conditions maintained on flame atomic absorption spectrophotometer. Limits of detection and quantification of metal along with certified and measured concentration were also noted. Statistical treatment of data, minimum, maximum, mean, median, standard deviation, standard error, skewness, kurtosis, correlation, and quartile distribution were also noted. Results and discussion. Basic statistical distribution parameter for the selected metal concentration in the hair of lung cancer patients are shown in this table. Most of the metal exhibited a large range of concentration as given by minimum and maximum levels. On the average basis, highest concentration was observed for sodium, followed by potassium and iron, while lowest concentration was observed for manganese as well as cadmium. A random distribution pattern was exhibited by most of the metal as demonstrated by high standard deviation and standard values on one side and distinctively dissimilar mean and median levels on the other side. Basic statistical distribution parameters for the selected elemental concentration in the hair of lung cancer patients are shown in this table. On the mean basis, highest concentration was observed for sodium, followed by potassium and iron, while lowest concentration was noted for manganese and cadmium. Maximum asymmetrical distribution pattern was observed for cobalt and lithium, as supported by large skewness and kurtosis values. Correlation study of selected metals in the hair of lung cancer patients are shown in this table. Metal data were also subjected to correlation study in order to find out the mutual relationships and are shown in this table. In the hair of cancer patients, bold values indicate a significant correlation among the selected metals such as iron, potassium, chromium, cadmium, chromium lead, which indicated communal variation in the hair of the patients. Correlation study of selected metals in the hair of healthy subjects are shown in this table. Similarly, bold values indicated significant correlation were observed among the selected metals such as potassium and iron, manganese and nickel, and chromium and cadmium. Red bold values represented the negative significant correlation among the selected elements such as lithium and sodium and Fe and sodium. Another important dimension of the present investigation is the multivariate analysis. Quantitative information regarding multiple relationship of these selected metals were assessed by principal component loadings. Principal component loadings of selected metals in the scalp hair of lung cancer patients is shown in this table. PCA, principal component analysis, yielded 5 PCs of the metal with cumulative variance exhibiting 89% of the whole data. PC1 yielded highest loading for magnesium, potassium and iron. PC2 yielded highest loading for copper and nickel. PC3 yielded highest loading for calcium, cobalt and cadmium. PC4 yielded highest loading for sodium and zinc. PC1 and PC2 were regulated by internal body metabolism and also controlled by environmental pollutants, particularly transportation related factors. Principal component loading of selected metals in the hair of healthy subjects is also shown in this table. In the hair of healthy subjects, PCA yielded 5 PCs of metals with cumulatively exhibiting 84% of the total data. PC1 yielded the highest loading for sodium, potassium, lithium, chromium and cadmium. PC2 yielded the highest loading for zinc and iron. PC3 yielded the highest loading for calcium. PC4 yielded the highest loading for magnesium and cobalt. PC5 yielded the highest loading for copper and nickel. It was also found that the loading pattern of selected metals was appreciably different as noticed in case of patients. Another important aspect of the present investigation is the comparative evaluation of selected metals based on demographic characteristics. Habitat-based variation of selected metals in the hair of lung cancer patient and healthy subjects as shown in this bar graph. The mean concentration of potassium, copper and lithium were found to be markedly higher and mean concentration of strontium chromium was lower in the hair of lung cancer patients as compared to healthy subjects. Comparative average concentration of selected metals in the scalp hair of lung cancer patients and healthy subjects based on food habitat is shown in this figure, which revealed that are in smoking patients than smoking healthy donors. Salient findings: most of the selected metals were randomly distributed, but randomness was comparatively higher in lung cancer patients. Mean levels of copper, cadmium, lead were significantly higher in the hairs of lung cancer patients. PCA and CA revealed dissimilar apportionment of selected metals in the cancer patients compared with the healthy subjects. Here is the last slide. Recommendations: body burden of other essential toxic metals should be explored. More clinical studies should be conducted, and future research should be focused on the relevance of respective mechanism. Acknowledgements. First of all, thanks to the organizing committee of this very special conference. I am grateful to the Department of Chemistry, University of Education. I am also thankful to the Head of Department, Professor Dr. Muhammad Abir Rashid. Thanks to my lab students and staff members, and very special thanks to the staff of Peenam Hospital, Faisalabad.
Thanks a lot, Mr. Muhammad Abdul, for sharing your research work. Here the session has ended. Now I would like to hand over this next session to Ms. Samia Zahid.